All right, I'll just get us kicked off here and then hand it over to our speaker today. Hello everyone, welcome to the IWGC webinar series. This is Eric Patterson at Michigan State University. Thank you so much for coming. Before we get started today, I wanted to announce that I need everyone to register for the IWGC newsletter. So go to the website. That's how you'll get information about upcoming seminars and webinars. We will have 6 other talks this calendar year, ending on December 5th. Our next one will be June 27th by Fabian Lamaga at Embrapa, talking about genome sequencing Ergospis plana. So everyone tune in for that one. That'll be the next one, and then you can hear about the one after that later. So with that, I'll turn to today's speaker, Diana Gamba, who will be giving her talk, Local Adaptation to Climate Facilitates a Global Invasion. So thank you so much for coming today. I look forward to your talk.
Thank you so much. Hi, everybody. I'm very happy to share with this audience our work on the causes and consequences of local adaptation in one of the most destructive invaders in North America. So today I'll be talking about Bromus tectorum, also known as cheatgrass, and hoping to have this paper out soon. Local adaptation to climate facilitates a global invasion. So we know that biological invasions are a key driver of global change, but the mechanisms fueling the worst invasions remain poorly understood. And in particular, the role of local adaptation remains unclear. Bromus tectorum is a great system for studying the role of local adaptation during invasion because it is native to Eurasia and Northern Africa, though it has a wide native range distribution. It was likely introduced repeatedly as a crop contaminant during the European expansion in North America in the late 1800s, and now it is present in all states in the contiguous United States, occupying different habitats and being especially detrimental in the sagebrush steppe of the United States Intermountain West, as you can see in these pictures. So in these ecosystems, it not only displaces native perennials, but also it has changed the fire regimes of these regions, making fires more frequent and earlier in the growing season. So as I mentioned, cheatgrass occupies many different environments, and we can see this in this map of the Western United States, where it has been reported in at least 20 different ecoregions according to the classification by the Environmental Protection Agency, including warm and cold deserts, semi-arid regions, and also plains. So it occupies that wide and diverse climates. And some characteristics that make cheatgrass a powerful system for invasion genomics is that it's an annual selfing diploid plant. So it's highly inbred, and it has a recently published high-quality reference genome, allowing genomic studies in detail. And I want to acknowledge that this project is part of a large collaborative and interdisciplinary group, the Bromcast Network. So this group is formed by experts in multiple fields of ecology, evolutionary genomics, rangelands management, and statistical model. And this group seeks to predict the trajectory of the cheatgrass invasion in Western North America under global warming. So my contribution to this project was to understand what is the role of genomic diversity and local adaptation during the invasion. So to get us started, biological invasions represent these shifts that can dramatically alter the genomic composition of populations in the invaded range. And a common prediction in invasion genomics is that diversity in a novel environment, here the invaded range, would result in diversity will be reduced in this novel environment due to colonization bottlenecks or genetic drift. So we can imagine a scenario of few introductions into the invaded range, where we would expect a strong pattern of local adaptation in the native range. So this genotype home advantage, so a wet genotype being having higher fitness in its home wet environment compared to the dry genotype and vice versa, but a very weak pattern of local adaptation in the invaded range. So this pattern of local adaptation can be described in terms of adaptive clines, and I'll be using this term quite often throughout the talk, in where this would correspond to a significant trait environment association. So in the y-axis, this is the trait genetic variation, or the genetic component of the trait variation, and how it changes across an environmental gradient. So we would expect a very strong correlation in the native range, consistent with strong local adaptation, but not in the invaded range. And this could be due to, as I mentioned, the low genetic diversity in the invaded range, but also because other processes like phenotypic plasticity could be behind the success of the invasion. However, we know that biological invasions are more complex because multiple colonization events could increase genetic diversity in the new range. And if environmental variation is similar between ranges, we would expect, or we could hypothesize, that adaptive clines will also be consistent between ranges. So this trait environment association is not expected to be perfect in the invaded range, because of the stochastic nature of where individuals end up during the invasion. So while some genotypes might have a trait value that is well matched to their new local environment, so for example, genotypes that are close to this line, there could be other genotypes that have a larger mismatch. So we would expect potentially a pattern of local adaptation in the invaded range, but potentially not as strong. So with this framework in mind, we sought to answer these three main research questions. First, do we see evidence of hygiene flow into North America? Second, are claims consistent between North America and the native range? Do we see evidence of selection maintaining those claims? And is local adaptation stronger where cheatgrass dominates? So to answer these, and with the help of dozens of seed collectors around the globe, we put together a diverse panel of 307 cheatgrass accessions. Today, I will focus on the invaded range in North America and the native range in Eurasia. So I grew these plants in a common environment at Penn State University. I got life history and performance phenotypes for a subset of 169 native and invasive genotypes. So I'll introduce these phenotypes later on in the talk. And then on the 307 genotypes, we performed whole genome sequencing, and this resulted in a dense genomic data set of around 15 million SNPs that I used for genome-wide association studies and also assessments of genetic diversity, and a filtered data set of around 250,000 unlinked SNPs that I used for population genetic structure analysis. So for the first question, do we see evidence of hygiene flow into North America? And here I'm going to show multiple analyses on population genetic structure to answer this question. So to start, here I'm showing results here I'm showing our sample genotypes in the native and invaded range. And the genotypes are colored by ancestral genetic cluster. So I'm showing 4 ancestral genetic clusters, which was the best number of supported clusters in the data set. And as we can see in the maps, all genetic clusters are present in Europe, and all of them are present in Western North America, indicating that a great amount of the native range diversity made it to Western North America, but not so much to Eastern North America. And this neighbor joining tree shows the genetic relationships between genotypes. So here I marked in gray places in the native range. So these are genotypes that came from the native range um, in here in this map. And in stars, I marked branches that contain genotypes in the invaded range. So we can see that invasive genotypes are closely related to many genotypes in the native range, especially from Europe, supporting that there have been multiple introductions through time. And looking at FST values, which describe population genetic differentiation, here I'm showing values between different geographic regions in the native and the invaded range. We can see that these values are lowest between invaded range regions and North, Central, Eastern Europe, supporting this idea that most introductions came from Europe. And when looking at values of genome-wide nucleotide diversity, which is a measure of genetic diversity. So here I'm showing these distributions of genome-wide nucleotide diversity on the x-axis in these two graphs in the native and invaded range. So we can see that in Eastern North America, genetic diversity is very low. And interestingly, in this region, cheatgrass is not considered problematic, and it mostly occurs at this ruderal plant that persists in highly disturbed environments. However, when we look at Western North America, genomic diversity, it's pretty high and comparable to the most diverse region in the native range, which is Europe. So again, supporting that a great amount of genetic diversity made it to Western North America from the native range. So we were also interested in understanding how genetic structure or genomic diversity is structured along space and environment. So first, here I'm showing results on the pattern of isolation by distance, or IBD, in the native compared to the invaded range. So we found a very strong pattern of isolation by distance in the native range, with genetic distance in the y-axis increases as spatial distance increases here in the x-axis. But in the invaded range, the pattern is more complex in that at very close distances, we see very distantly related genotypes. So very high genetic heterogeneity at small spatial scales. But also we see these groups of almost clonal genotypes, so genetic distance almost zero, occurring at close proximity, but also all the way to over 3,000 kilometers. So this shows that there's been very long distance gene flow, potentially within the invaded range. So we see that isolation by distance is not strong in the invaded range, but we were also interested in understanding how the environment shapes genetic diversity in both ranges. So we conducted these variance partitioning analysis to analyze the contribution of the abiotic environment and spatial distance to the explained variation of total genomic variation. So these Euler plots, the square represents the total genomic variation, or the area of the square represents the total genomic variation, and the circles represent the contribution of the abiotic environment and spatial distance. And the abiotic environment here comes from multiple bioclimatic variables that I used for these variance partitioning analyses. So as we can see in the native range, both the abiotic environment and spatial distance explain variation, genome-wide variation. But in the invaded range, only the abiotic environment is important, suggesting that even though the isolation by distance pattern is very messy, genotypes are sorted along different environmental gradients. And I'll go in more detail with this idea. But I wanna bring your attention to these groups of almost clonal genotypes, we found pretty interesting results in terms of these almost clonal groups. So these almost clonal groups are genetically distinct groups, so they occur in different parts of that neighbor joining tree that I showed at the beginning, and they're nearly half of these of the North American accessions are within one of these near clonal groups, and each of these groups are seemingly very successful in a different part of its range. However, in this principal component analysis, I'm showing the distribution or in how the climate space of genotypes in the invaded range. And we can see that these clonal groups occupy most of the climate space in the invaded range. And they're also differentiated by environment. So from a Permanova analysis using environment, using clonal group to predict the environment, we see that each group occupies very different environments. So the distribution of these clonal groups shows that dispersal is not limited because they can reach very far distances, but it appears that environmental filtering is important because, as I mentioned, each group occupies very different environments. So these clonal groups are contributing to a pattern of local adaptation in the invaded range. However, we were interested in understanding how genotypes in general are sorted along different environmental gradients. So for these, I'll just go into the second question, where we asked if clines are consistent between North America and the native range, and if we see evidence of selection. So here, now I'll present results on our growth chamber phenotypes. So this principal components analysis summarizes the phenotypic variation across the different genotypes. And here I'm coloring them by their ancestral genetic cluster. So those K equals for genetic clusters. And PC1 represents this life history axis of late flowering and great vegetative investment to early flowering and high fecundity. And to some extent, this axis is also correlated with ancestry in that the pink genotypes are mostly on the early flowering, high fecundity side. So when comparing, and these genotypes are both in the native and invaded range. So when comparing invaded and native range genotypes, we find very consistent trait environment clines between ranges. Here I'm showing an example for PC1. So in the y-axis, there's PC1 from these principal components and how it changes with maximum vapor pressure deficit. And maximum vapor pressure deficit is a measure of aridity, and I chose it in this to test for this client since it's one of the variables that best explains variation across the climate space of cheatgrass globally. So we found in general earlier flowering and higher fecundity in drier regions, both in the invaded and native range. And these p-values come from a linear mixed model. So these models include a kinship matrix representing relatedness between genotypes as a random effect. So the fact that they are significant indicate that selection is likely driving or maintaining these clines because the pattern is independent from the genome-wide pattern of variation represented by that kinship matrix. So this is evidence that selection on aridity gradient is driving variation consistently in the invaded and the native range. However, selection could be acting on any of the correlated traits in PC1. So we were interested in testing for selection, particularly in flowering time, since it's an important variable that describes life cycle, especially for an annual plant like cheatgrass. So to this end, as I mentioned, this is part of a large collaborative group where we perform different common gardens in different sites in the Western United States, trying to cover all the different climates that cheatgrass occupies in the Intermountain West. So these common gardens were replicated over 2 years with fall plantings and spring harvestings. So for this analysis in particular, I chose the two most contrasting common gardens. One is this low elevation hot dry site, and then the other is this high elevation cool and wet site. So we compared flowering time and fitness between these two sites over 2 years, and we find that the results are pretty striking. So here on the y-axis, I'm showing measurements of fitness. So for 2022 represented by mean seed count, and for 2023 represented by mean inflorescence mass. These two are highly correlated. So in both years, we find that in the hot side, early flowering has a greater advantage over delayed flowering in both years. And then in the cool side, delayed flowering is more advantageous. So this provides strong evidence that changing selection along temperature aridity gradients maintain a life cycle climb in the Intermountain West. And this is also consistent with our results for flowering time in our common environment at Penn State, so in a growth chamber, in that we see earlier flowering potentially being selected for in drier regions. And I wanted to bring back, here I'm showing days to flower and how the consistent climbs between the invaded and the native range. So we performed a genome-wide association study in flowering time because we were interested in understanding the genetic basis of flowering time variation. So I'll show results of this Manhattan plot represents the results for the genome-wide association study that we use this univariate mixed model with random kinship effect, so also correcting for relatedness or population structure between genotypes. And interestingly, we find this very large haploblock in chromosome 1. So these results require further validation. But what's interesting is that this haploblock is enriched for gene ontology terms related with seed embryonic, post-embryonic, and fruit development, and also seed dormancy. So it seems that this locus is important for life cycle in general and potentially responses to environment changes during seed development. And even though it requires further validation, it would be interesting to see to what extent structural variants are also important for local adaptation in cheatgrass. But even though we need more validation for these locus, I want to show here the patterns of variation in the native and invaded range for the top GWAS NIP in our GWAS. So this NIP, it's in this position in chromosome 1, and it's close or downstream of this ERDJ3B gene, which is related with his stress tolerance during seed development. So we can see the allele distribution in the native and invaded range. And when comparing the patterns of variation or of allelic variation between ranges, we see, again, consistent patterns between the native and invaded range. In that the, the pattern of flowering time variation is the same. And then when we look at how the alleles vary in terms of climate, so here in the y-axis, I'm showing again maximum vapor pressure deficit. We can see that the late flowering alleles are more common in more humid regions, consistently in both ranges. So this suggests that even the specific mutations underlying local adaptation are repeated between the native and invaded range. And now I want to show, I'm going quicker than I expected. So we'll see. So for my third question, is local adaptation stronger where cheatgrass dominates? For this question, we applied, so a common motivation for understanding the drivers of local adaptation is to use this information to forecast how populations will adapt to future environmental conditions. And in this case, these future environments or future environmental conditions could be translated into environments in the invaded range for an invasive species. So we use this genomic prediction approach to predict genotype environment matching or offset in the invaded range. So one assumption is that in the native range, and I guess our results also support this, that in the native range, local adaptation is very strong. This is because populations have had more time to evolve in those native local environments. So we can use we can use a predicted optimum based on genotype environment associations to see how genotypes in the invaded range are far or close away from this predicted optimum. So the genetic distance between the native predicted optimum here and the observed genotypes in the invaded range would correspond to this genomic offset measurement. And again, this predicted optimum was obtained from a genome environment association study in native ecotypes using representative climate variables of the global climate distribution of cheatgrass. So now I'm going to show results of these offset measurements. And so this map shows the distribution of those offset values in North America, with the purple values corresponding to lower genetic offset, so greater and genotype environment matching, greater genotype environment matching. So what we can see is that genetic offset is usually lower in higher latitudes, but also in general in Western North America. And it's actually significantly lower in Western North America compared to Eastern North America. This comes from permutation tests. But we were particularly interested in the Great Basin and offset values in the Great Basin, not only because cheatgrass, this is one of the most heavily affected areas by cheatgrass, but also because there's been a lot of studies in this region, including this great study that performed many field surveys around like 11,000 sites in the Great Basin, and then they calculated or quantified percent cover of cheatgrass in all of those sites. So we use this data to ask if offset is lower where cheatgrass dominates. And here are my main result, or a very striking result, where we find that indeed lower offset, that offset is much lower where cheatgrass dominates. So this is a very strong pattern. So here in the x-axis in the box plots, I'm showing the cheatgrass abundance in the Great Basin of low and high abundance meaning observations with 15% or more of cheatgrass cover. And these high abundant sites do correspond with greater or lower genomic offset. So this is great evidence that genotype environment matching promoted, it's greater where cheatgrass dominates, and so it has promoted invasive spread. And invasive spread, in turn, could allow more opportunities for cheatgrass to disperse and colonize new environments. So this also points out that these regions of high cheatgrass abundance are also potential sources of locally adapted alleles that could keep dispersing to similar environments and continue the invasive spread of cheatgrass in still untouched environments in Western North America. So now, to summarize my findings in my research questions, we do see, so for the first question, is there evidence of high gene flow into North America? Yes, but we also find evidence of very high gene flow within the native range, in that we see those almost clonal genotypes being far-flung, reaching almost over 3,000 kilometers across North America. However, even though we see high gene flow into North America, we also have evidence of environment playing an important role for structuring the genomic variation of cheatgrass in North America, and consistently with their native range. And are clines consistent between North America and the native range? Do we see evidence of selection? Yes, as I showed, we find these consistent clines of, in general, life history and flowering time. And we find that in Western North America, temperature aridity gradients are potentially maintaining this cline of earlier flowering being more advantageous in drier regions, and later flowering more advantageous in more humid regions. And then is local adaptation stronger where cheatgrass dominates? Yes, with our genomic prediction approach, we find that genotype environment matching is lower, or sorry, it's greater. So genomic offset is lower, where cheatgrass is more dominant. So this is interesting because it allows us to make this link between local adaptation and ecological dominance, which is a common prediction that local adaptation should increase population size and abundance at a given site. So we find evidence of that prediction. So some take-home messages. We find evidence of pre-adapted genotypes that enabled invasive local adaptation. So basically, the native range diversity that made it to the invaded range is being reused in a very similar fashion in the invaded range, probably because the environment gradients in North America are similar to those in the native range. So those genotypes matched the environment in the invaded range very well. Again, in Western North America, temperature gradients maintain a life history cline. But in Eastern North America, genotypes are low diversity and potentially ruderal. So this is interesting in that we see these very strong patterns of local adaptation in Western North America, but not in Eastern North America. And I didn't go into much detail for results in Eastern North America, but none of the clients that we tested for when we compare Western and Eastern North America, none of those clients are significant in Eastern North America. Probably because of the low diversity and also because the life history strategy is different, in that is more ruderal. Again, we see this greater genotype environment matching where cheatgrass dominates. And in general, I think that this is the overarching message would be that understanding the drivers of local adaptation is critical for predicting and managing invasions. This is especially, I think it's important because most of the effort for invasive species is focused on the environments where cheatgrass has invaded, so in whether there's ecosystem invasibility or not, but not so much on the role of genomic diversity of the invader. And in this case, we see that any new sources of genetic diversity could keep reshaping local adaptation in the invaded range, for example, in response to global change. So it's important to consider stopping incoming introductions that are still being potentially introduced as crop contaminants. I think that is potentially a big issue for cheatgrass still. And also in terms of hygiene flow within the invaded range, there should be more attention to regions where potentially cereals or wheat is exported from one state to other states. Make sure that we're not sending out seeds of cheatgrass with those crops either within North America. So with that, I'd like to thank the Lasky Lab at Penn State University, where I've done all of this work. It's been a really great opportunity to just learn a lot of things about analysis of local adaptation and working with genomic data in an invasive plant like cheatgrass. And I also want to thank all the people who helped with assisting with different aspects of the project, including all the co-authors that a lot of them provided seeds. So they went out of their way to collect all these seeds and take notes and follow a very strict protocol that we asked for the seeds to be collected. And yeah, this ended up being shorter than I expected. Yeah, I went too fast, but with that, I would take any questions and I can go back to any slide that people are more interested in seeing, maybe. But yeah, that's all I had for today. Thank you.
So you don't get to hear the applause, but you get to hear me speak. So yes, if anyone has any questions for Diana, please type them in the Q&A section, and I'll read them out loud. And then Diana, maybe you could answer them for us.
Sounds good.
Yeah. I'll get us kicked off a little bit while people are writing or whatever. So you mentioned the importance of possibly the importance of structural variation, right? So you're talking about general, diversity very generally, and in this case, you're measuring a lot of things like single nucleotide polymorphisms. What is the role, or what do you think the role of structural variation is, as opposed to something like SNPs, for generating novel variation in these invaded ranges? Or is that still an area that we don't really know kind of the relative impact of like structural variation versus these kind of classical SNP diversity?
Yeah, so yeah, all of the genomic data that I worked with was based on SNPs. And so my suspicion for structural variation being potentially important comes from this result where we find this big haploblock. This is not unique to the invaded range. So both native and invaded genotypes have this sort of variation. So I don't think that is something that occurred de novo in the invaded range. But in general, structural variants are important for maintaining variation that can be locally adapted because they reduce recombination between without crossing. Like if there's outcrossing, then these structural variants won't there won't be recombination between them, so they maintain all of those genes linked together that could play a role in local adaptation.
I didn't think about that. That's very interesting.
Yeah, so I think that's the main reason why people are interested in studying structural variation and its role in local adaptation. It's because they allow maintenance of those genetic variants, even with there won't be recombination between those blocks. But for a selfing plant like cheatgrass, recombination is expected to be, I guess, inefficient. So potentially structural variants are not necessary. But even though outcrossing is very rare, it is possible.
Mhm.
So in that sense, they could be important in if there's some outcrossing between different genotypes.
Interesting. Okay. Yeah, I'm
Yeah,
coming from a very mechanistic worldview, molecular worldview, where structural variations can have big impacts on individual traits like herbicide resistance and this sort of thing. So I'm thinking of it as, a way to generate novel genetic diversity or a novel phenotype that can be selected upon. So the very, very mechanistic view.
yeah, and I think that's why this type, this locus in particular requires more work and more validation
Yeah,
just to get to more of those mechanistic understanding of what is the role of this, other than explaining variation in flowering time.
sure. We have a question from Dr. Todd Gaines at Colorado State. He says, fascinating work, Diana. I agree. Is there any indication of differences between crop field and rangeland populations, whether in phenotypes or in genetic structure?
That's a really interesting question, and that's something that we haven't really addressed in that when looking at genotypes in the invaded range, we considered different ecoregions, and then we did this eastern versus western comparison, but not we didn't look into land use, for example, and how that can impact genetic variation. So that's definitely something that would be worth looking at in how those different land uses have affected genetic structure, genomic diversity in general. So yeah.
Yeah, just to piggyback on that too, especially in a species that's not outcrossing and doesn't have a long distance seed dispersal mechanism, you may get lots of recurrent selection in even smaller regions, like in a field or something like that. Very cool.
Yeah. Yeah, and it's interesting about the dispersal of cheatgrass in that generally plants shatter, like the fruits just shatter and the seeds fall off to the ground. But they do have these awns that are kind of like very pointy, and they can get into fur of cattle, for example.
Mhm.
So they could potentially be dispersed long distances through cattle. Uh,
Mhm.
but I think that the instances of very long distance dispersal represented by those clonal groups that I showed might be more. I'm not sure what is the mechanism behind that long distance dispersal, but I think it's potentially
Yeah,
human movements.
interesting. Any other questions from our participants? So if there is no other questions, oh, Todd again, is there anything you would change about how you sampled or your genotypes?
Um, so I mean, I think that including more samples in the native range would be interesting because we're potentially still missing some of the environments of the native range environments, like those in Northern Africa. Cheatgrass is native there too, and I think more sampling in the native range would be interesting.
Mm.
In terms of sampling in the invaded range, so we were interested in understanding local adaptation. So we prioritized sampling different sites to sort of increase that environmental variability between sites that we sampled. But I think it would be really interested to see within sites genotype multiple individuals within a site to better understand to what extent there is genomic diversity at a very small distance. From what we see at very short distances, especially in Western North America, you can find very distantly related genotypes, but it seems that they are occupying different environments too. But within a site that is one environment, it would be interesting to see what are the genotypes there, what is the genomic diversity at a small scale.
Right. So I just wanted to mention this will be recorded and available to everyone. So we will be posting this soon.
Great.
And everyone, if you have questions to follow up with Diana, you can always email her and get in touch.
Yes.
If that's it, I think we'll let you go, Diana. And thank you so much for the talk today. We really appreciate it.